So our last speaker in this session is Dr. Salman Qasim. He is an assistant professor from Rutgers University. Salman and I met each other a couple of years ago when we co-hosting the Nano Symposium at SFN about a human intracranial research. And we have after like happy hour, like a party right afterwards. And we started with 6 panel speakers, but after half an hour, it becomes like 20 people. I saw like I probably drink too much, have double visions, but apparently just people in the field start growing. The reason I mentioned this because I think that level of engagement or the level of arousal also reflecting what someone was studying. So basically, he's going to tell us today the cells and the circle level representations of arousal in the human brain. Thank you, Salman.
No. Excellent. Thank you, Jay-Z, for the nice introduction. Thank you, Tawfiq, for the invitation here. It's been such an incredible session to see so much work across scales and across species, and I hope I can add just a little bit to that. Now, I'm going to tell you about the work from my lab investigating human arousal at the level of cells and circuits, but I'm going to start from a slightly different place. I'm going to start with memory because I am a memory researcher. All of my work stems from this basic principle that the sad truth about life is you forget much of it. But while this sounds sad, I think it highlights what is a pretty universal aspect of human experience, which is that out of 1,000,000 experiences, there's some endogenous mechanisms that allow us to highlight specific instances for memory. So, for example, I've been to Toronto more times than I can count, but the experiences that stand out to me are the ones that mean something, like I get to see my grandma. So, why is it that there are certain experiences that stick out more? The question that my lab asks is, what neural mechanisms strengthen memory for relevant events such as this? Now, why does this matter? At a basic level, understanding the answer to this question can highlight, again, those endogenous neural mechanisms for building strong memories. But this is also important because it goes wrong all the time. It goes wrong in the case of maladaptive tagging, which you might see in post-traumatic stress disorder, when an event becomes maladaptively too strong in memory. And it goes wrong in memory loss, when emotional or important memories that should be strong fade away. So in my lab, we come at the question of these mechanisms of memory strength from a few different angles. We use lab-based memory tasks to investigate the psychophysics of memory. We use psychiatric symptom assessment to see how these interact with self-report psychiatric information. We use computational models of cognition to infer latent computations we can't observe. And as a neurophysiologist and bioengineer, we use direct brain recordings and the resulting time series that we extract from these recordings to try to identify informative biomarkers of the different behavioral metrics that we're measuring. Now, we call this approach cognitive electrophysiology. You've heard a bit about it from Drs. Fu and Petkov, so I won't beat you to death with these direct brain recordings, but I want to go into a little bit of detail about the method here. Now, obviously, in research like ours, we get the opportunity to leverage a very direct recording of in vivo brain activity from neurosurgical patients who are undergoing clinical mapping for epilepsy. This results, as you've heard, in the ability to record mesoscale local field potentials, as well as from the tip, or sometimes I guess from the shaft of these probes, single unit activity, so firing rates from single electrodes. But it's important to ask what the virtue of doing this type of invasive work is. You know, these are human subjects undergoing a difficult time in their life. And what we get out of this, the reason it's worth doing, is we get action potentials, this fundamental unit of complex computation. We get to see how they might act together at these larger scale local field potentials. And these two signals provide us high spatial and temporal resolution compared to what we might get non-invasively. But as the other speakers have also mentioned, we get the opportunity to get in the loop and causally manipulate these biomarkers. And so for us, that gets us closer to translational techniques from the basic science upwards. Now, to return to the scientific question, my lab is interested in what neural mechanisms strengthen memory for relevant events. And I'm going to operationalize relevance in this specific case as having to do with affective significance. Now, that's a simplification. There's a lot more that makes memories relevant, and my lab studies some of those things. But I think it's very naturalistically true for a lot of people that events that tap into our emotions feel relevant and we remember them quite well. So we're going to talk about two studies in which we look at the cells and circuits that track emotion information for memory. So to begin, I want to talk about how we measure emotion information for the duration of this talk. Now, I did my PhD in spatial navigation, where it's very easy to measure things in space. There's metrics. What kind of metrics can we use for emotion information? So one candidate measures emotion information in 2 dimensions, valence and arousal. It's a simplification. It's a model that allows us to reduce the complexity of emotional experience into 2 principal dimensions. Valence, capturing how negative to positive an occurrence is, and arousal, capturing how boring to engaging an experience is. I'm hoping for some of you this talk is somewhere in the top right of this 2D representation. Now, the useful thing about this psychological model is it's intuitive, but it also generalizes and replicates fairly well across cultures, and it provides us a simple 2D metric for emotion information. And this 2D space lets me cheat a little. It lets me lean on my experience analyzing neural correlates of space. So just as a review of what those are, there's very, very stark neuronal correlates in especially rodent models of physical location in the form of place cells capturing activity in a single location, or grid cells which show activity patterns that map entire locations with a tiled grid. Now, aside from being very stark neuronal correlates of experience, the reason that these are useful to us is because they let us theorize about how they might support different mental computations. In our case, we're interested in inference, so the ability to, for example, in physical space, compute a shortcut. In non-physical spaces or in abstract space like emotion space, that can be very useful. So in this first project, I'll talk about work I did in collaboration with Dr. Lukas Kunz in Germany, where we formulated an idea to assess whether neurons in the human entorhinal cortex map 2D emotion space. So we used a fairly simple free recall task to begin with, where subjects see images that are drawn from a database of emotionally variable images. They do a little distractor task, and then they just freely recall the images in whatever order. Now, we pulled these images from a database where we could control the valence and arousal features of these images, and we end up with a 2D emotion space of 240 images of roughly that shape. And we can look at the influence of different image features on subsequent memory. There's a lot of image features that predict memory. For example, people really remember images with the presence of an animal in it, but arousal is also a strong predictor of subsequent recall. And I'll remind you that these are crowdsourced arousal and valence ratings, yet they predict something about our patient's behavior. So the next question we want to ask is, do they predict something about our patient's brain activity? So to that end, we're recording from 19 patients across 30 sessions. We're recording hundreds of neurons across medial temporal lobe regions, including parahippocampal cortex, hippocampus, amygdala, and entorhinal cortex. And neurons in these regions are pretty responsive after the presentation of the image cue, and they respond to a lot of things, just like memory was influenced by a lot of things. But I'm going to zoom in on 2 features that are particularly interesting to us, which is first image arousal. Here I'm just plotting the example activity from one neuron, where we see that it increases its firing rate for high arousal images compared to low arousal images. And we find a lot of neurons like this. They're primarily located in parahippocampal cortex and entorhinal cortex. We also see examples of neurons that are encoding valence. So by that I mean they're changing their firing rate more for one particular valence than the other. In this example, positive valence is encoded with a weaker firing rate than negative valence. And these valence encoding neurons are primarily located in the amygdala, where we might expect them. And this was a nice sanity check that the crowdsourced ratings do seem to induce some sort of alteration in neural activity. But we're not interested in this like univariate approach. What we're more interested about is the tuning of neurons in this two-dimensional valence arousal space. To get at that question, we take each picture, we take the evoked firing rate for each picture, and we assign it an arousal and valence, and we bin that firing rate and arousal valence space. So across 240 images in a session, we get firing rate maps of each neuron across that space. We can compute what is called a grid score from the autocorrelogram of that spatial firing rate, and then we can do a lot of shuffling to make sure that the grid score that we're recording is significant related to like null shuffle data. When we do this, the output is for each neuron, we get a sense of how grid-like its spatial firing pattern is in this two-dimensional valence arousal space. And what we find is primarily these grid-like neurons are located in the entorhinal cortex, just as we'd expect from rodent data in the physical world. I'm showing you one example of an entorhinal cortex cell here. The firing rate map is in the top left, where red dots indicate the fields where that neuron is more active. Right next to it is the autocorrelogram, where you can hopefully see some periodicity. And above we have the waveform density. And we can show you quite a few entorhinal cortex neurons like this. We recorded 31 of them in total across our 19 subjects. And this is cool, but it could be a weird statistical quirk, right? We want to make sure that the pattern we're recording here has some correspondence to rodent grid cells in physical space. So one of the traits that we were most interested in was, do these grid cells lock to the ongoing entorhinal theta phase, like rodent grid cells are very well known to do? And the short answer is that they do. So I'm showing you examples of spiking in these entorhinal grid-like cells aligned mostly to the trough of the ongoing theta phase. And a simple comparison we did was to compare phase locking to the cells that are just encoding arousal or just encoding valence, but not both. And these grid-like neurons do exhibit higher phase locking than those. And again, mostly focused around the trough of theta. But what I'm really interested in is memory, right? And we want to see if the grid-like activity differs as a function of memory, which is a question that's very hard to answer in the rodent data. Now, during encoding, what we observed was that grid-like neurons exhibited what we call a subsequent memory effect. Their firing rate increases for images that were later recalled versus those that were forgotten, but only if those images were in the firing field. Similarly, during free recall, when they have to, just freely say whatever images they remember, right before they vocalize their mnemonic response, we see a similar pattern. We see these grid-like cells increase their firing rate, but only if the image had appeared inside one of the firing fields during the encoding period. So, which you see here. So there's some evidence then that these grid-like neurons not only encode this kind of relational, two-dimensional emotion space, but seem to modulate the firing rate as a function of memory processes. Now, I'm not showing you a Herculean amount of supplementary analyses we did to to validate the existence of these spiking patterns and their specificity, but I'll summarize some of them for you. Grid-like neurons were not driven by image content. They were not driven by a separate 2D semantic space or 2D visual space itself, by changes in occupancy or sampling across subjects. They're stable across time and across session. And most interestingly, we didn't find place cells. That's a big open question and one I think that is really worth discussing. So hopefully if you find me, we can talk about it. So in summary, we asked, how does the brain represent the relationship between items in this 2D arousal valence space? And the answer seems to be that neurons in the entorhinal cortex can form a cognitive map of emotion space. So we're going to scale up now here. We're going to expand our scope beyond single neurons to the mesoscale LFPs that enable us to track broader biomarkers and maybe even the relation to neuromodulation. So as is the theme here today, I'm studying this in humans with intracranial electrodes, but now we're using a huge sample. This is 148 subjects that were recorded as part of a massive multi-site DARPA project in which participants performed a similar task, a verbal free recall task, instead of an image-based free recall task. And we're recording here primarily from those macro scale electrodes now, not the microwires, but in amygdala and hippocampus. Again, we're crowdsourcing emotion ratings for the words in this task, so we can assign each word an arousal and a valence. And again, these crowdsourced arousal and valence ratings are predictive of memory, particularly arousal. You tend to remember words that were more engaging than not. So now again, we can ask, what brain activity underlies enhanced memory for these arousing words? And the focus here will not be on single neurons, it's going to be on the LFPs, and in particular, the high frequency activity, which not only is thought to index ensemble spiking, but as has been previously shown, is deeply involved in memory, and Jay-Z has shown is deeply involved in aversive image viewing. So we thought it a good candidate signal for the intersection of emotion and memory. So in this one trial I'm showing you here, during encoding, you see activity from a single hippocampal electrode during two words. One word is jail, the other word is purse. Both words are subsequently to be remembered, but you see a big power difference. The reason for that is one word is high arousal and one word is low arousal. But when you look at the same electrode for a different pair of words that was both subsequently not remembered, they weren't encoded, you don't see that difference. If we pick an example amygdala electrode, the same pair of words, we see the same difference, where high frequency activity seems to track with arousal, but only during successful memory encoding, not during failed memory encoding. Now we can look at this over all of the hundreds of electrodes in all of the subjects. So what I'm plotting here is just the probability of recall as a function of high frequency activity, but only for low arousal words, we don't see any relationship. For medium arousal words, we don't see any relationship. But for high arousal words, all of a sudden, high frequency activity in both amygdala and hippocampus seems to track with the probability of recall. Now that's a nice correlation, but we're trying to get beyond correlations here and ask something a little more causal. Do affective disorders unlink this relationship between high frequency activity, emotion, and memory? Now, a subset of patients in this cohort provided depression and anxiety ratings to give us the opportunity to do a pretty crude parsing of more depressed versus less depressed patients. But crude or not, we find that depressive patients performed worse at the task overall, but specifically exhibited a behavioral phenotype that was super important. They're specifically impaired at arousal-mediated memory. So whereas arousal was beneficial to memory in our healthy cohort, that benefit goes away in our more depressed cohort. And as that benefit for arousal-mediated memory goes away, we also see that the relationship between high-frequency activity in the hippocampus and amygdala and arousal-mediated memory also goes away. Now, this is, again, just another correlation. What we wanted to do next was something a little more truly causal, which was to use direct electrical stimulation to try to, again, unlink these relationships. So 27 subjects performed a variant of the task in which we applied direct high-frequency stimulation to hippocampus and also surrounding medial temporal and temporal cortex regions, 2 on, 2 off, 2 on. And you can see the effect of that stimulation on the LFP. It saturates. And what we find is that stimulation, again, selectively disrupts memory for higher arousal words than lower arousal words. A very similar pattern to what we just saw in depression. And this is only hippocampal stimulation. Medial temporal and temporal cortex stimulation does not do this. So finally, we can look at the neural basis for this potentially by comparing the post-stimulation period and the pre-stimulation period. And we find that hippocampal stimulation selectively disrupts HFA, but these surrounding control regions, when stimulated, do not have the same effect. So in summary, we're finding single-cell correlates of this cognitive map for arousal and valence in the entorhinal cortex, a broader mesoscale signal for arousal-mediated memory in the hippocampus and amygdala that is disrupted by both depressive symptomology and direct electrical stimulation. The hope is we can take advantage of these biomarkers to potentially stimulate some of these regions, maybe get in the loop and disrupt maladaptive arousal-mediated memory in PTSD. I'll take 5 seconds to make a pitch for the underexplored phenotypic relevance of epilepsy for these types of neuropsychiatric explorations. Refractory temporal lobe epilepsy is very comorbid with a lot of different psychiatric disorders. It moves this group beyond just a convenience sample. So if we identify nice behavioral phenotypes like depression-specific disruptions to arousal-mediated memory, we can focus on those phenotypes in these populations. But only 21% of epilepsy centers do comprehensive neuropsych eval. So hopefully that's a thing we can improve. And with that, I will thank my funding sources, my collaborators, and the patients who participated in this study. Thank you.
Thank you for the wonderful talk. I am a little bit confused in the part where you map the grid cells in your emotion, in your valence, arousal space. The way that people map grid cells in Broadens requires moving through space. So you have to move in a continuous, well, that's where Broaden does, trajectory through space. Beth Buffalo described grip cells in monkeys, and it's also through eye movements. But in your case, did they move through the space that you designed? So how does the whole parallel that you put between, I mean, I like the idea, but I cannot, because there is a definition of a space, velocity. I mean, grid cells are supposed to do that.
Yeah.
In your case, how does it work?
That's a great question. Our analysis is more akin to picking up the rodent and moving it from position to position, which is obviously not naturally how they move. But the reason we thought this was kind of ethologically similar was a thing I'm not showing you here is that one of the ways in which free recall is thought to function is through this memory clustering and memory search. So items that are close together in either semantic context or temporal context, or in our case, emotional context, tend to cluster together in recall. And what that means is you can actually track recall trajectories. So movement from items that are close together in emotion space, where you tend to recall them in sequence, and then move in sequence to items that are close together in another aspect of emotion space. So ethologically, we think that the way recall might be functioning is kind of in this continuous trajectory. We're simply not able to record it. So we have to do the next best thing, which is take these transportations and try to make a map out of it.
And your prediction is that they're going to move from one to another state faster if they're closer.
Yeah.
I mean, that's a testable prediction.
Yeah, and it is a tested aspect of the behavior here. Is the subjects show significant clustering in recall for items that were close together in time, close together in semantic meaning, and close together in emotion space.
For you, incredibly impressive work. I really enjoyed it. For the depressive symptoms situation, am I getting this right? Is it important that they place similar valence on the images as the control group?
That is a great question. So this kind of depression, selective, diminishing, could it be that they assign the same arousal and they simply don't care, like an anhedonic response, or that their assignment of arousal is different itself? That we can't answer at this point, but I honestly, I believe it could go either way. It's like a 50-50 question to me. And I think it also depends on the depressive symptom at play. We're taking very broad measures of, how depressed are you? A more granular approach is going to look at, are they more anhedonic versus are there other, kind of more expressive depressive symptoms, to try to answer that question.
Maybe I can ask a question now that I have the mic. So this hippocampus is traditionally not associated with arousal. Where does that signal come from?
Yeah, I have been getting very interested in the notion of locus ceruleus just pumping the medial temporal lobe with noradrenergic input. And now that's a thing I can't test because we're not measuring from locus ceruleus, we're not measuring neuromodulators. But my kind of pet theory, my hand wavy theory, is that this is coming from locus ceruleus. There's really nice recent work demonstrating that both noradrenergic and dopaminergic terminals in the hippocampus receive direct termination from locus ceruleus. So that would be my pet guess.